Well, good morning, everyone. I'm really thrilled to be here. I appreciate the organizers who have given me this opportunity. Um, First of all, I want to let you know that even I'm going to talk about the retina today and some of the work we've done with metabolomics, but that even retina biologists have a difficult time explaining the retina to each other because it is a very highly sophisticated structure, both physiologically and cell biologically. And in fact, Richard Maslin, in his recent review, wrote that Darwin might not have been able to write on random selection if he had actually known how the retina functions because it's so elegant. But the retina is found in the back of the eye along the innermost periphery, and it's composed of a laminar structure of multiple neurons and glial cells. And if you imagine that the lens is at the top, the light actually comes through all of the layers of the retina and impinges upon the rods and cones, which are shown at the bottom of the slide. The rods being the photoreceptors that absorb light under dim conditions, and the cones being important for color vision and daylight vision. Now this slide just shows a random list of retinopathies, some of which you may have recognized, such as age-related macular degeneration, retinitis pigmentosa, and diabetic retinopathy. And the point that I really want to make here is that the causes of retinal degeneration are both genetic and environmental. Okay, so diseases such as macular degeneration have a large environmental component, even though we recognize that there is a genetic predisposition. Retinitis pigmentosa, on the other hand, is completely inherited, and what's important to understand about all of these, really all of the retinal degenerative diseases, is that there are no effective therapeutic interventions that can cure these diseases. And so understanding mechanism is really, really critical. And if we look at genes that contribute to retinitis pigmentosa, we can see that there are genes that are autosomal recessive, autosomal dominant, X-linked, and there are many, many different kinds of genes. Some of these genes are actually involved in the phototransduction pathway itself that responds to light. Some others are trafficking genes like myosin-associated genes, ciliopathies, and so forth. And you can also see in the grayish-blue sections that there are a lot of forms of RP in which there are unknown genes. But one of the prevailing wisdom ideas is that oxidative stress and metabolic dysregulation are two causes that might be common to different RP disorders, no matter all of these very, very different genes. Okay, and why this might be true, it might require understanding a little bit about the retina. So one of the interesting things about the retina is that relative oxygen consumption in the photoreceptors themselves is higher on a per gram basis than any other tissue in the body. And that the energy and oxygen consumption is higher in the dark and the light. So you have all of these different factors that are interplaying. And high levels of polyunsaturated fatty acids are required for the structural development of photoreceptors, and that this is a highly energetic process. And also, interestingly enough, light is a risk factor for retinal degeneration, particularly UV light, which can create reactive oxygen species. And this has sort of been verified to some extent by papers such as those from Dr. Sepko's group, showing that NRF2, which is a transcription factor that regulates a lot of antioxidant genes, can promote neuronal survival in neurodegenerative diseases of the eye, which she was looking at. And that antioxidants of various types can slow photoreceptor cell death in various models of retinitis pigmentosa from genes that are functionally unrelated. So we decided to use a mouse model for retinitis pigmentosa called the RD10 mouse, which is a defect in one of the phototransduction genes. And as you can see here, RD10 is susceptible to light. On the left, in the top panel, you can see there is a lot of degeneration in the light and less in the dark reared. And in the bottom panels, we've actually done time courses so that we could select a time point at which we could look at early events in retinal degeneration. And so in the light-reared mice, that would be somewhere between day 14 and day 20. And so in collaboration with the wonderful people at RTI, we took an unbiased metabolomics analysis approach, and we raised wild-type or RD10 mice under cyclic light or in the dark. And after 17 or 18 days, we also exposed them to bright light for 3 hours or dark adapted them overnight. And then we removed the retinas, froze them, and passed them on to RTI. And this is just a workflow from them where they do UPLS, MS analysis, and then a statistical analysis, and then look for library matches in their own in-house databases or the HMDB. And since mouse eyes are really small, you have to understand that this in itself was an issue. And Suraj Dugana, who actually did this work, was able to get a signal response from only 10 milligrams of retina extract. And you can see here that the low abundance region has really good signal to noise activity. And this is what actually allowed us to perform these studies. So if you look at a PCA analysis, what you'll see right away in the RD10 retinas with the red and the black dots is those were raised in cyclic light. And they differentiate, they segregate completely from retinas from mice that were raised in the dark. And this is distinct from wild-type retinas where there seems to be less of a differentiation between the light and the dark-raised animals. If you look at OPLS-DA analysis, if you look at paired analysis between each of these different groups, and this is where it gets a little bit complicated for us. We may have overdid ourselves with all the different things that we did. If you look at wild-type mice in the top two panels, you'll see there is a difference between mice raised in the light and versus dark and light adapted. And on the right side, raised in either cyclic light or in the dark and dark adapted. And the same is true for the RD10 mice. You see pairwise significant, highly significant differences under both of those conditions. Okay. Now, if you look between genotypes, so you look at the RD10 versus the wild-type mice, you also see very distinct differences between the mice, no matter what the conditions, whether they were raised in cyclic light and incubated in the dark before euthanasia, or whether they were raised in cyclic light and incubated in the light and raised in the dark and incubated in the dark. And so this suggests that there are, this validates the fact that metabolic profiles are different in both wild type and mutant mice if you incubate them in the dark versus in the light, or if you raise them in cyclic light versus in the dark. So obviously all of these profiles represent metabolites. And I'm just showing two here from this analysis. It's early days for us in taking a look at this, but you can see from these tables, which are organized by VIP in RD10 versus wild type, there are the usual players like glutathione, oxidized glutathione, and a lot of lipids. But one of the things that we noticed early on is that there seems to be a difference in acylcarnitine accumulation in the mice. And acylcarnitine accumulates between the inner and the outer mitochondrial membrane as part of the carnitine shuttle, which translocates fatty acids from acyl-CoA in the cytosol to acyl-CoA in the mitochondrial matrix. And the significance of this is not really understood yet, but it certainly represents an intriguing aspect of our work. the other thing that I would like to make a point of is if you add in all of the compounds, not just the ones that matched the databases, you can see that there are a large number of unknowns with very high VIP value. And my colleagues at RTI told me that this level of unknowns is really unmatched in the other tissues that they have studied. That there's about a 20.9% difference in the left panel, an 18.2% ratio of known to unknown in the right panel. And this is pretty much true throughout the 7 pairwise comparisons that we did. So in conclusion, what I'd like to say is that these results demonstrate for the first time using an unbiased broad spectrum analysis that retinas from RD10 and wild-type mice have distinct metabolic profiles. PCA analysis demonstrates clear separation between and dark-reared RD10 mice, while there's not really as much separation in the wild-type mice. The pairwise comparisons do suggest that there are distinct differences between the different light conditions for both the wild-type and the RD10 mice. And also that there are unusual numbers of unknown metabolites in these profiles that might not be defined in metabolic databases to date. And also that light induces changes in the level of acylcarnitine compounds compared to dark, particularly in comparison of wild-type and RD10 mice. And these results are consistent with the observation of faster degeneration in the light in the RD10 mice. So we have some future directions. These include identifying some of the unknowns with high VIP values. I think that would be a tremendous benefit to the vision field and perhaps to others who study neurobiological diseases. To determine which cell types in the retina where these group differentiating metabolites are localized, because remember, this is a complex tissue, and the glia interact with the neurons. It's not really clear which cells these metabolic changes are occurring in, and obviously identify pathways. And also, we'd like to treat RD10 mice with compounds that have been shown to delay retinal degeneration and determine whether that influences the group differentiating metabolites that we've are identifying in our studies. And finally, I'd like to acknowledge the people in my laboratory at UNC Chapel Hill, as well as the wonderful people at RTI, MRC, who have done all of this work and teaching us about metabolomics. And I also appreciate the grant funding both to myself from the NIH Common Fund and also to the RCMRC. Thank you very much, and I'd be happy to answer any questions.
Any questions?
What is the sample here? On which sample you did metabolomics? Which sample? Part of the eye?
So we dissected out, I'm sorry. So we dissected retinas from the mice, either in the dark or in the light. And we used dim red lights in the dark. And we had 4 retinas from 2 mice each that we pooled in a sample. And then we had 5 separate samples for each analysis.
So you have taken only tissue?
Yes.
Because there are fluids also, like aqueous humor, vitreous humor.
Yes, yes.
You have taken only tissue?
Yes, we have taken only tissue to the extent that we know. What we do is we take out the retinas and we immediately drop them in liquid nitrogen. And so there may be some aqueous and vitreous humor that comes with the retinas. We haven't been able to control for that, but that's a good point.
Yeah, the biological conclusions that you are drawing, that carnitine shuttle may be important in this scenario. suppose if we want to extrapolate this study to humans and for a live person to study the retinal tissue is not possible. So how you see this? How we can go with this? Is there any other sample source with which we can study and we can know some idea?
You mean in order to figure out whether or not these approaches will work in humans? Is that what you're asking? Well, that's obviously that's the million-dollar questions, because it's very difficult to get samples. Post-mortem samples are really not appropriate for these kind of studies. Primates are not really used for these kind of studies anymore. So I think the best thing for us to do is to define the metabolic pathways, understand what compounds may retard retinal degeneration in the mice, and then establish clinical trials to see whether or not that occurs in humans. I mean, for example, light is known as a factor that causes retinal degeneration. So physicians are always telling their people, don't use your eyes. I mean, wear sunglasses and so forth. So I think what we're focused on is the ability to define pathways, and that will allow others to define therapeutic agents, which then can be tested in humans.
Thank you.